Welcome back to the second speaker of today's seminar, Mamie Wang from Yale. She's a PhD student in the computational biology section at Yale University, working in the lab of Steve Kleinstein, and her research interests involve developing and applying computational methods for understanding B-cell specificity and repertoire biology during the human immune response. Looking forward to your talk.
Thank you for the introduction. Let me quickly share my screen. And good. Hello everyone. Today I'm presenting my work on benchmarking embedding methods, including protein language model on predicting antibody properties. As we know, antibodies are key components in our immune system to detect and eradicate invading organisms, and what makes antibody particularly fascinating is their ability to recognize their target epitope on the antigen with high specificity. This helps our immune system to fight pathogens effectively and also makes them attractive as drug candidates. Recently, there have been growing interest in training antibody language model for different predictive applications on antibodies. Here are some of the examples that I know. These models are trained on large data set of BCR amino acid sequences and can be used for a variety of tasks. For example, imputing missing residues and predicting paratope location and also predicting antigen specificity. And most of these recent language models are based on a neural network architecture called BERT, which was originally designed for natural language processing tasks developed by Google. And it's known for its effectiveness in capturing long-range dependency in sequential data because of their use of a self-attention mechanism that basically weighs the importance of each of the words in the sentence when the model is encoding its meaning. And BERT model was originally pre-trained. The training process of the BERT model is usually separated into two processes. First, pre-training, and then pre-training was basically performed on a large data set of text data using self-supervised learning objective. For example, MLM, masked language modeling, which are predicting masked words in a sentence. And in the case of antibody language model, it's predicting masked residues within a sequence. And for the downstream application, you can basically, one method is directly using the embedding, which are the numerical output from the last hidden layer of the pre-trained model, and simply use these embeddings to downstream supervised machine learning model for different prediction tasks. In addition, more formally, you can also fine-tune BERT on specific tasks with small amount of task-specific label data. And basically, in the first part of my talk, I will discuss my effort in evaluating the direct use of embedding in predicting receptor property. And then this work was recently published in Nucleic Acid Research. For the second part of my talk, I'll discuss my recent research in fine-tuning these pre-trained model on antigen specificity prediction task. So here are the first part of my project, and here's the outline of it. So in this project, I was particularly interested in profiling different embedding methods to examine if they capture biological information of the antibody sequences, including sequence property and receptor binding. I'm curious as to if there are any differences in what type of biological information these different type of embedding are good at. So in short, there are many traditional ways to represent the sequence in numerical value as embedding. And then one very simple way is simply compute the amino acid frequency given one sequence. And also there are other methods, for example, using Blossom substitution matrix, which is based on the likelihood of amino acid get substituted with another amino acid based on multiple sequence alignment studies. And there are also factors that are physical-chemical based on the amino acid. And I also examined one model that are called in Virtu-Vac model, which is a shallow neural network that are trained to predict the neighboring context of amino acid given one target chimer within the sequence. And finally, I'm also examining 3 different protein languages model. 2 of them are trained on protein, including ESM2 and also ProT5, and the other one, anti-BERT, are trained specifically on the BCR sequences. So in terms of the data set, I collected paired single-cell BCR full-length data set from different public data sources. In total, there are 1 million B cells with both the heavy chain and light chains. And for the binding specificity task, I collected most of the data from the CoVAP database, which curate public data set from different publications. And we also have some sequences from a collaborating lab based on ELISA tests on a monoclonal antibody sequences to test against the SARS-CoV-2 spike protein binding, and the labels are binary. So here is a quick UMap visualization of the different embeddings that I tested. The first one is a BCR language model, and these two are the protein language model. And this is the word2vec model, and this is the two traditional encoding, the frequency and physical chemical encoding. And then individual color here represent the V gene usage and the J gene usage. Just by looking at the plot, all the embedding encodes information on the gene usage. And I performed the same visualization on the somatic hypermutation frequency and junction length as well, and it seems to encode the information as well pretty good. However, to formally evaluate how well each embedding encodes this property, we need to do something more complicated. Here. So basically, we were training a supervised model given the embeddings and evaluated using nested cross-validation. And in short, the inner loop for the nested cross-validation is used to select hyperparameter. For example, in this case, there is a regularization parameter in the support vector machine classifier that we use to predict the properties. So basically tune this part of the parameter within the inner loop. And for the outer loop, it's going through the test data set and then compute the final test prediction performance. And the training test validation split have no overlap between different samples or subject in our data set. And I also, as a random control, I randomly shuffle the labels to the specificity data set so that we can use it as a random baseline and then run through the same procedure here. And here's a box plot for the nested cross-validation test set performance. And then I'm showing the F1 score here. And the higher it is, the higher the accuracy for the prediction. And individual box here showing different tasks, including given the embedding, predicting the V gene identity, and then J gene family, and then isotype. And this is the same for the light chain. And the first three columns here are the protein language model, and then the last two are the word2vec and also traditional embedding. And we see that across different tasks, the best performing models are the protein language model actually. And the gray dots here are for the random baseline, and all of those embedding perform better than the random baseline here. And similarly, I performed the same procedure for the sequence regression test, which including predicting the somatic hypermutation and also the junction length here. And the evaluation metric here is RMSE, and the lower the value is, the better the embedding R's. And also we noticed that all the language models here have a lower RMSE compared with the basic encodings here. And it's better than the random baseline. And finally, I evaluated the embedding on the receptor specificity prediction. In terms of this data set, we obtained around 15,000 sequences and that have binary labels to whether it's binding to coronavirus spike protein or not. And then we try to balance the data set by sampling some of the repertoire that are collected pre-pandemic so that it's roughly 50% binding and 50% non-binding sequence within the data set. And I evaluated the use of different sequence input. For example, because we are using the single cell BCR data set, we can have the full-length paired heavy and light chain sequences, but we can also subset it to only the heavy chain full-length sequences and the CDR3 heavy and light chain pairing and CDR3 heavy chain only data and then input these sequences into the model and then generate an embedding to evaluate the performance on predicting the binary binding status to SARS-CoV-2 spike protein. And we noticed that the longer sequences input we give, the better the performance overall is it. And basically, for the best performing models that are using the full length heavy and light chain pairing, antibody performs the best among the models, even among the protein language model. And also one interesting thing is that even though immune-to-vac model, which is a word-to-vac model that are trained on BCR sequences streamers, are simpler using simpler architecture, it's actually performing slightly better on average compared with the larger pre-trained protein language model here. And in this case, across all the comparisons, the best model performs around 79% nested CV balanced F1 score, which is antibody model trained on full-length pair sequences. And then I will move on to the second part of my talk, which is trying to evaluate how does fine-tuning can help with predicting the binding specificity. And then in this case, I tested on two different antigens. The first one is same as the previous work, the SARS-CoV-2 spike protein. And the second one is predicting on influenza HA. And for fine-tuning the model, we gather the data, the labeled data specific to these antigens, and then basically initialize the weights using the pre-trained model, and then train it again on the supervised test to predict whether it's binding or not. And after training this model, we also compared it with our previous benchmarks that are using the model embedding plus the SVM approach and see if it's fine-tuning improves the performance there. In addition, for validation, we also apply the specificity classifier on individual sequences for the repertoire that are undergoing immune response to SARS-CoV-2 vaccination and also influenza vaccination to see if there's any signal of immune response that get captured for the classifiers. So here are the 4 different antibody language models that are publicly available that we can download the weights from, and then it's arranged by increasing size. So the model are anti-BERT, anti-BERTA, and then these 2 models are from the recent paper from Brini lab, which are trained also on the paired BCR sequences, and then they also fine-tune the ESM model on BCR MLM tasks. So the first step is applying the benchmarking using the pre-trained model embedding, and then train an SVM on the pre-trained model embedding. And this example slide is showing for the SARS-CoV-2 prediction data set. And basically it's the same plot from the previous slide, except I'm adding more benchmark antibody models here. And I'm showing the cross-validation AIRR-C here. So the best performing model is the fine-tuned ESM2 model on the BCR MLM task. And also we noticed that we also tested the performance on the ESM model originally, and then find it's worse than after fine-tuning the ESM2 model, interestingly. And the next step is taking all these models, all the 4 models here, and then perform the supervised fine-tuning on predicting the SARS-CoV-2 spike protein prediction. So the left bar here are exactly the same plots showing on the left panel here. And then the right corner are the corresponding model that are undergoing fine-tuning. And we see that the performance improves after we do the fine-tuning across all the 4 different antibody languages model. And we tested the Wilcoxon-Ranson test and then find that the differences are significant. Also, individual lines here are corresponding to the cross-validation folds exactly. And we are trying to investigate what happened after fine-tuning, and previous studies have shown that for protein language models that are trained on antibody sequences, it learns to focus more its attention on the CDR region, and this is associated with improved specificity prediction. So to evaluate the effect of fine-tuning the antibody language model, we just compute its self-attention activation by averaging across the layers and heads. And we do so by sampling 50 random antibodies that are specific for SARS-CoV-2 spike protein and influenza from the training data set, and basically compute this average attention across so in this case, this is the heavy chain, and the axis is the position along the heavy chain, and the y-axis are the difference of activations, attention activation between the fine-tuned and pre-trained model. And the dotted line here are the mean of these differences. And the region here, the 3 bars here are showing the CDR region. And we notice that there is an increase of average attention activation across all the 4 models after fine-tuning in position corresponding to the CDR region. And this is particularly pronounced in, for example, the CDR3 region. And finally, to further evaluate whether the fine-tuned language model classifier can capture the specificity information, we apply our fine-tuned classifier to a single-cell BCR repertoire dataset that measures the immune response to SARS-CoV-2 vaccination and influenza vaccination. And for this slide, I'm only showing the result for SARS-CoV-2 mRNA vaccination. And basically, it has 1 time point that are sampled 1 week after the second booster vaccination, and then this is the day 28 plot here. For the control sample, I take the pre-pandemic repertoire and assume that it has a lower level of SARS spike protein specific sequence, and just intuitively, the control sequences should be predicted have a lower score, predicted have a lower probability of binding to the SARS-CoV-2 spike protein. And I basically just, for each of the sequences within the repertoire, I apply the classifier and then compute the predicted probability of binding and take the average across all the sequences within the repertoire. And I find that after vaccination at day 28, the predicted probability is much higher compared with the control sample, which is consistent with our speculation. Just for a summary, we show that fine-tuning a pre-trained antibody language model can significantly enhance the specificity prediction for the SARS-CoV-2 spike protein. And also we see a similar trend for the influenza hemagglutinin. And we notice that there is an increased attention on the CDR region after the fine-tuning, which probably indicates that it captures relevant feature for the antigen specificity. However, there are also a lot of limitations for our study. One limitation is the data, because the training data, for example, for the influenza viruses are quite small, and also they are not very balanced between the binding versus non-binding sequences. So we have to sample from other control repertoires to train the classifier, and it's potentially limiting the general reliability of the evaluation. Also, our focus on the antigen are limited because of also the data limitation, and we only focus on the SARS-CoV-2 spike protein and influenza HA because of the data availability. And also, we because of the label are simply binary on the antigen level, and so it does not distinguish between different epitope within the antigens. Finally, it's the attention mechanism is quite complicated to interpret, and because of the model complexity. And then in summary, I want to thank my dissertation committee and also acknowledgement for my lab mates. And thank you for your attention. I'll take any question next.
Thanks so much. This was really nice. Are there any questions from the audience? I'm going to start. You showed that after fine-tuning, there was more attention in the CDRs.
Yeah.
Yeah. And then in your summary slides, you said that you don't know the epitopes of these antibodies, I guess. So to what extent does this tell us? So do we know which of these residues are important for binding and not important for binding? Could we be even more specific in what this in what you're showing there?
Yeah, for the attention, it's actually I'm taking the average across the layer. So it's like
Yeah,
for each of the attention, it's dependent on the previous layer. So it's not exactly correspond to exact location on the antibody sequences.
I
So
see.
it's really hard to interpret that.
I see. So how do you interpret then that we see more attention in the CDR regions? Is that a good thing? I
Mm,
guess it's a good thing.
yeah, it's focusing more on the CDR. I guess there's more diversity on the region.
Mm, mm, mm. Yeah. Tim?
Yeah, I thought the analysis of the peripheral blood was super striking. If you can go.
So this one, the
Yeah,
repertoire analysis.
yeah, I was wondering here, is the data independent? Like, is there any training data that goes into this, or is it separate?
So for the fine tuning, it's using a separate data set from this analysis. And for the control, the day 28 sample are from the plasma blast of the after vaccination. So you would expect there are a lot of spike protein specific. And also for the repertoire data, I explicitly exclude those sequences that appeared in the training data set that are within one clone of it. So I think it would be independent.
Cool. So is it different people
Yeah.
than the training data? Oh, cool. Uh, how wait, how do you do the plasma blast, or what's the is it from peripheral blood, or it's from or
It's
how
free
do you
from
do that?
peripheral blood.
Okay. Cool. Thank you. That's amazing.
Felix?
Oh, hi. Yeah, very interesting. I was wondering, in your summary, you mentioned data quality, the importance of data quality, and you mentioned quantity, and you mentioned needing the binding, more binding data, more receptors with binding data. But I guess two questions. When you think of quality, what other quality, what other characteristics are you thinking about? And then it seems like you took data from many different studies. Did how much did you have to worry about the different protocols, the sequencing protocols versus the bioinformatic protocols, or did you reanalyze the data, or how did that work for you?
Yeah, actually for the specificity data, I just directly take it from the public data set. And for the other sequences, I'm actually filtering only for the single cell BCR sequences because they tend to have lower sequencing error compared with other data set, for example, box. So that's basically limiting my data like availability to train the receptor specificity because most of the data are bulk and then some of them are also not full length.
Okay.
Would you go back to slide 17?
Yeah.
So here you're showing that we can see differences between control and day 28 using protein language models on the repertoire level. Is that what you're showing? And do you also have controls where you do use super simple methods like difference in V gene usage? So how surprising is that do we see this also with more traditional approaches, or do we only see these differences with protein language models? Maybe you showed this,
Yeah,
or maybe.
actually didn't test for the simpler like VGene usage, but I tested on using just the pre-trained model embedding without doing anything. It also shows the difference.
Yeah, could be nice to also use very simple approaches. That's true.
Yeah,
Because protein language models are so high powered, and then people could ask, why do we need them if we can just see the differences very easily with more simple yeah.
that makes sense. I will try to do the analysis with a
Yeah.
simple method.
Yeah, just any more questions? This was really nice. Thanks so much again. If there are no more questions, then we close. And please come back next month. Next month, we have a focus on germline gene polymorphisms, another very hot field. So yeah, thanks so much, everyone. Have a good day or evening. Bye-bye. Bye-bye.